Lecture No. 11. Krebs cycle. The aim of the lesson: formation the knowledge about the Krebs cycle. Objectives of the lesson: to learn the discovery of the Krebs cycle, to distinguish the stages of the citric acid cycle. 3. To explain Krebs cycle steps. This cycle is named after Sir Hans Krebs, an English scientist who discovered it in the 1930s. In 1939, A. Chibnall proved that the Krebs cycle is carried out in plants. In 1953, Hans Krebs and F. Lippmann, for the discovery of the role of coenzyme A, received the Nobel Prize for their work. Krebs cycle take place in the mitochondrial matrix. Acetyl groups, 2C, are involved in the cycle, joining the 4C compound, oxaloacetic acid, OAA, as a result of which citric acid, 6C, is formed. The cycle is therefore called the citric acid cycle, and the tricarboxylic acid cycle because it is these acids that participate in it. The Krebs cycle, known as the citric acid cycle or tricarboxylic acid, TCA, cycle. The citric acid cycle is the final common pathway for the oxidation of all biomolecules, proteins, fatty acids, carbohydrates. This cycle is important as it provides electrons/high-energy molecules to the electron transport chain for the production of ATPs and water. Pyruvate formed at the end of glycolysis is first oxidized into acetyl-CoA, which then enters the citric acid cycle. Aerobic respiration involves 4 stages: glycolysis, a transition reaction that forms acetyl coenzyme A, the citric acid, Krebs cycle, and an electron transport chain and chemiosmosis. Krebs cycle steps: oxidative decarboxylation of pyruvate to acetyl-CoA. Step 1. Condensation of acetyl-CoA with oxaloacetate. Step 2. Isomerization of citrate into isocitrate. Step 3. Oxidative decarboxylations of isocitrate. Step 4. Oxidative decarboxylation of alpha-ketoglutarate. Glycolysis and the citric acid cycle. The oxidative decarboxylation of pyruvate, end product of glycolysis, to form acetyl-CoA, initiator of Krebs cycle, is the link between glycolysis and the citric acid cycle. In the conversion of pyruvate to acetyl-CoA, each pyruvate molecule loses one carbon atom with the release of carbon dioxide. During the breakdown of pyruvate, electrons are transferred to NAD+ to produce NAD, which will be used by the cell to produce ATP. In the final step of the breakdown of pyruvate, an acetyl group is transferred to coenzyme A to produce acetyl-CoA. Formation of acetyl-CoA through the transition reaction. Through a process called oxidative decarboxylation, the transition reaction converts the 2 molecules of the 3-carbon pyruvate, C3H4O3, from glycolysis into 2 molecules of the 2-carbon molecule acetyl coenzyme A, acetyl-CoA, NAD middle.H+, and 2 molecules of carbon dioxide. 2 pyruvate plus NAD 2++, 2 coenzyme A yields 2 acetyl-CoA plus 2 NAD plus 2H++ 2 CO2. Enzyme citrate synthase aconitase isocitrate dehydrogenase alpha-ketoglutarate dehydrogenase succinyl-CoA synthetase succinate dehydrogenase fumarase malate dehydrogenase 1 NAD are formed from a molecule of pyruvate in the oxidative decarboxylation of pyruvate to acetyl-CoA. Steps of Krebs cycle. 1. Acetyl-CoA and oxaloacetate condense, forming citrate. Enzyme citrate synthase. Citrate is isomerized to isocitrate by a rearrangement of the molecule. Enzyme aconitase. 3. Isocitrate is oxidized to alpha-ketoglutarate. Enzyme isocitrate dehydrogenase. 4. Alpha-ketoglutarate is converted to succinyl-CoA enzyme alpha-ketoglutarate dehydrogenase. Steps of Krebs cycle. Continuation. 5. Succinyl-CoA is cleaved to succinate. Enzyme succinate thiokinase, succinyl-CoA synthetase. 6. Succinate is oxidized to fumarate. Enzyme Succinate dehydrogenase. 7. Fumarate is converted to malate by the addition of water across the double bond. Enzyme, fumarase. 8. Malate is oxidized, regenerating oxaloacetate and thus completing the cycle. Enzyme, malate dehydrogenase. What is the purpose of the Krebs cycle? The purpose of the Krebs cycle is the complete oxidation of glucose, resulting in energy-rich molecules that later produce ATPs in the electron transport chain. The citric acid requires oxygen as the cycle operates only under aerobic conditions, as NAD+ and FAD can be regenerated from their reduced form in the mitochondria only by electron transfer to molecular oxygen. The following are the enzymes that catalyze different steps throughout the process of the citric acid cycle. Citrate synthase aconitase isocitrate dehydrogenase alpha-ketoglutarate succinyl-CoA synthetase succinate dehydrogenase fumarase malate dehydrogenase. Conclusions: cells obtain ATP from breakdown of glucose in the absence of oxygen, as in glycolysis. Most organisms normally are aerobic and oxidize their organic fuels completely to CO2 and water. Foodstuffs feed into the citric acid cycle as acetyl-CoA, and the acetyl-CoA is oxidized to carbon dioxide and water in order to generate energy. The generation of energy from glucose is the oxidative decarboxylation of pyruvate to form acetyl-CoA under aerobic conditions. The cycle also serves in the synthesis of fatty acids, amino acids, and glucose. Electron transport chain. It is a chain of protein complexes located in the mitochondrial membrane. This chain carries electrons split off from donor molecules. Protein complexes of the electron transport chain: NAD dehydrogenase, cytochrome BC1, cytochrome oxidase ubiquinone, and cytochrome C. ATP synthase. Each turn of the cycle also gives one ATP molecule. Each glucose molecule requires 2 revolutions to oxidize. At the end of the cycle, oxaloacetic acid is regenerated and can now attach a new acetyl group to itself. D and tricarboxylic acids of the Krebs cycle are given in the order of their formation, from oxaloacetic acid, citric acid, isocitric acid, alpha-ketoglypteric acid, succinic acid, fumaric acid, malic acid, oxaloacetic acid. 612 6 plus 62 greater than 62 plus 6238. Thanks for attention.